Thanks, Sean and Janan. This has been a fantastic meeting. I've gotten so much out of it. It's really great to be here. So I'm going to start today centering my argument on a conventional piece of wisdom said by physicists and biologists alike, which is that physics is easy and biology is hard. This gets said in different ways, but the statement I'll make is that people say physics is relatively compact and simple compared to other disciplines. And biology is unimaginably complex and fundamentally irreducible. So just to give a sense of how this gets stated in different ways, here are quotes from a biologist and a physicist, respectively. Dawkins is saying, biology is the study of complicated things that give the appearance of having been designed for a purpose. Physics is the study of simple things that do not tempt us to invoke design. Now, the purpose of that argument is clear, given the book and his predisposition. But again, he's using this conventional contrast to invoke that. And then Murray Gell-Mann, here for a very different purpose, is saying the science of biology is very much more complex than fundamental physics because so many of the regularities of terrestrial biology arise from chance events as well as from fundamental laws. Right now, he goes on to use this to talk about why we need complex systems, why we need new types of science, why we should study biology in more interesting ways, and why we should invest in it over the next 100 years. But again, he's saying something about why biology is fundamentally more difficult than physics. And so the argument I want to make is that this is really about questions. It's about the types of questions and motivations we have in different disciplines. And in physics, what we happened to do as time progressed was start off with really easy problems and really easy questions, and then move towards the hardest problems and hardest questions. And in biology, we did the opposite. We started with the hardest problems, with the hardest questions, and that became the focus. And so the question really is, as time goes on, are there easier problems, easier questions to return to? What sorts of new traction does that give us? Where does that lead us? And why haven't we seen these yet? And that's what I'll spend the rest of today talking about. So just to give a sense of what I mean by starting with easy or starting with hard problems, physics sort of started here, right, with an interest in planetary motion. And now while discovering how this worked required a triumph of technology, observation, new types of mathematical theory, and so forth, and a lot of very smart people working very hard, at the end of the day, the final set of equations, the final description, just isn't, in the grand scheme of things, that complicated, right? Showing Kepler's laws, which this is a pictorial representation of, to a mathematician is sort of laughably simple to them. And so at the end of the day, this turned out to be a really easy question with a rather easy answer. And biology instead started here, trying to ask, how do we explain this diversity? Right? How do we understand every single individual species? How do we understand the vast diversity of all these species, why they're so different, and so forth? And what I'm showing in this picture is roughly one size scale of a very narrow taxonomic group of just diatoms. And yet, even in this picture, the morphological diversity sort of baffles us. If we looked inside of these cells, the chemical and physiological and metabolic diversity that we'd see would also baffle us. And typically, as biologists, what we come to say then is the only way to understand a species is through some exceptionally detailed, highly contingent, very complicated historical evolutionary trajectory that leads us to one species that may never occur again in a replaying of the tape on our own planet or on any other planet in the universe. And for some questions, this is true. There are certain bizarre traits of organisms. We can sort of only answer why they exist in this context by saying it's about this detailed evolutionary trajectory. But I want to highlight that this is true only for some observations, questions, scales, problems. I'll turn this around in a second and show the types of questions and observations that we can ask that take us away from this perspective of only these highly contingent evolutionary trajectories. Van Savage and I wrote an essay about this idea where we say it's really about questions, it's not about disciplines. It may not even be about phenomena, it may just be about what questions you ask of those phenomena. So here we say, but perhaps there's no such thing as an easy or hard discipline. Maybe there are only easy and hard questions. Biology only seems so hard because it has been defined by a set of very hard questions. Physics only seems easy because centuries of effort by deeply insightful thinkers have produced a set of answerable questions, right? And in this, we produce this sort of pictorial representation of these ideas. There's many ways we could have cast this, but we say basically, you can think about different types of spectra of problems, right? You have a single mechanism operating in a system. Think Kepler's laws, one gravitational force, planets orbiting a star. Or do you have mixed mechanisms, the layering of multiple types of physics, so forth and so on? And then are you trying to make average coarse-grained predictions about something, or are you trying to make incredibly detailed predictions about specific events? So a great example is the stock market. Classically unpredictable. If anyone could predict it better, they would make untold sums of money. And the reason it's so hard to predict is because the number of layered mechanisms leading to it, from the physical dynamics of the climate affecting agriculture to all sorts of game theoretic decisions made amongst investors. There's sort of so many mechanisms interacting with it. And then also the thing that people are interested in predicting for certain types of high frequency trading is what's going to happen in the next second, the exact state of the market in a very detailed way in the next unit of time. It's an incredibly hard problem, and that's why it's so hard to predict, so forth and so on. But there are easy questions in this space for sort of any discipline. And I want to mention just quickly that physics has hard problems as well, right? So as physics has moved away from only studying fundamental physics and trying to understand prediction of different phenomena where there are lots of layered physical dynamics, think the sun and all of its various overlaying sets of physics. If we're trying to forecast the position of the next solar flare, that's an incredibly hard problem that requires this huge observational network of different ways to observe the sun, different satellites we've put in space. And the same is classically true for understanding the climate system or fluid dynamics or all of these different problems. So imagine if physics had started with trying to predict streams rather than planets, we might be in a very different space with how easy or hard or compact we think the discipline is. And this is just to highlight that one of the recent Nobel Prizes in physics was given for this complexity idea, recognizing that there are truly hard problems. Now, I don't want to say that biology doesn't have aspects to it that make it fundamentally more challenging. But I think we should see it something like this, overlapping spectra of the type of questions and problems we're interested in. So physics certainly has these hard domains like forecasting the sun or our climate system. And biology has these very hard problems about understanding individual species and particular evolutionary trajectories. But the question is, how far can we push biology into the easy domain? And so I just want to note that biology is a new type of emergent phenomena, of a new type of discipline. It is a complex adaptive system, and we should think that we need new perspectives, epistemologies, and ontologies, right? So I'm not saying that's not true. Biology will offer all of those to us as we continue to study it, and already has. But I also want to say that the underemphasized aspect of biology, the easy part, is that there are powerful coarse grainings and simplifying questions that are underdeployed. And I'll spend the rest of my talk talking about those. So what is one of these coarse grainings for a perspective like this? One way to make this simpler is to zoom out and ask questions about all of life. And here we start to see more law-like phenomena. So this is a wonderful picture from James Brown et al. This came out of work done by Jeffrey West, Brian Enquist, and Jim Brown. And this is the review article associated with that work for his MacArthur lecture. And what we see here is that if you plot the logarithmic mass of an organism against the biomass production rate, so this is how many grams of biomass you produce per year per individual. It's a measure of metabolic rate in sort of a normalized way. We see a roughly single relationship that goes across all of these different scales, right? We can put a vast amount of diversity on a single plot, going from single cell plants, algae, all the way up to multicellular animals. And so to a physicist, this looks like a law-like behavior. And I'll explain in a second where these come from. And the way we reconcile sort of that detailed perspective and this law-like perspective is by recognizing that at any one scale, we still have this vast amount of diversity, this huge amount of scatter that can be up to an order of magnitude in size, right? So this is where all of those details I was mentioning before live, but if we zoom out enough, we get a sort of easy answer to biology that puts all the diversity on a single law-like curve. We understand where these come from. It's really evolution operating under physical constraints, right? And so one way that we can think about this is take any trait of interest where you have some performance of that trait, either its capacity not to break like a bone, or some amount of energetic efficiency that leads to reproduction. And then you think about that as a function of body size, where you have some physical constraint that may dominate at one scale and leads to one of these scaling laws as an evolutionary optimization. And then maybe you at some point switch to some other physical constraint, or at all of these scales, you have the overlay of multiple physical constraints. But our argument is that you can write this down as an evolutionary cost function, which we do in this paper, and then simply the evolution's messy dynamic that's able to sort of hill climb and optimize these constraints over long periods of time. And so this is why we expect to see these relationships, but only under a few conditions. The same constraints have to apply to organisms of vastly different size, and the traits have to be independent enough of each other, right? So if you have traits that depend on really different constraints, and those two traits depend on each other, that can lead to messiness that won't lead to scaling. But the places where we have been successful in finding these scaling relationships relate to this first relationship, where we see the same constraints operating across lots of scales on organisms in a dominant way. And here we see a case where exception proves the rule. If you zoom in on the smallest forms of life, we see shifts in these scaling laws as you go from single cell bacteria to unicellular eukaryotes to multicellular life. And we've written a lot about what constraints are shifting as you go across these scales and why these architectures have to evolve in response to those shifts. And so this really sort of tells us that, in fact, we would see the counterfactual of if you shifted constraints, you should get different behavior, because indeed at the small end, we do shift constraints and we do see different scaling, and we have this one-to-one derivations that connect these two. What's interesting is we can take this and use it in all sorts of ways to predict other phenomena. So for example, you can derive the growth rate of an organism as a function of size and these metabolic rates. And here we then now get added predictions where in the theory, these asymptotic behaviors show up. And these asymptotic behaviors tell us where certain forms of life become fundamentally limited, right? So at the small end of bacteria, you predict a point where growth rate goes to 0. That should tell you the size of the smallest possible bacterium. And at the large end of unicellular eukaryotes, again, you have this point where growth rate goes to 0, mostly because the repair costs in both cases are taking over the cell. And this tells you the scale at which you should go from single cell life to multicellular life. And indeed, that's where you stop seeing lots of large single cell organisms and you start seeing small multicellular organisms. So this theory is not only good for predicting how the laws will change under different constraints, you can then use those constraints to make higher order predictions about the history of life, about the evolution, the major evolutionary transitions as life got more complicated, as basically a response to infinitely hard constraints that happen at different scales. Now, this is just to show that a lot of these predictions hold up in the same way that we would want different sorts of cosmological predictions to hold up. So we here put out a prediction for the smallest bacterium. It was smaller than the world record holder at the time. And then a few years later, Jill Banfield's lab found these even smaller new world record holder bacteria. And you can see that this roughly agrees with our average prediction for this smallest scale, right? And this is just coming out of a very simple energetic theory. So this gives us a lot of confidence, right? And this can be used in all sorts of ways. For example, if you have a putative fossil in a meteorite, as we did in the 90s, you can use the size of those to compare to this scale and rule in whether or not you think that might be a living fossil or not. We've also used these ideas to zoom even further into the cell, and this will rhyme a lot with what Nick was saying a few days ago. But you can take these same sort of law-like perspectives and say, once we have this energetic bound, once we have these growth rate dynamics, what does that tell us about the detailed dynamics of the cell? So we can look inside at sort of what's happening with all the chemical reactions, the macromolecular dynamics. And here we've been able to do things like predict requirements for what's known as the central dogma in biology, which is just basically the main way that you go from information to function. And here, I'm not showing any of the mathematics, but we can show how this theory leads to a prediction for how many ribosomes, this basic machine that turns genetic information into folded functional proteins, how many you need as a function of size. And again, you predict an asymptotic behavior where you would need more ribosomes than can fit inside the cell simply because all the dynamics are speeding up and you can't keep up with that. So we think this is really nice. It tells us now why you need this transition to unicellular eukaryotes. And we think that in bacteria, we have strong bounds for both the smallest organisms and the largest organisms. So not only do we understand an architecture of life, we understand where its boundaries are. I'll mention that you can now start to play games at sort of the grand evolutionary scale and say, well, what if life had discovered a much better or much worse ribosome? And in this forthcoming paper, we've been able to show that if you had a ribosome that was 40 times slower, you couldn't encapsulate the type of cellular life we have in any meaningful way. So given the types of protein concentrations you need in the cell, if the ribosome were much slower for its size, say 40 times slower, you can't encapsulate life at all. And if the ribosome were 10 times faster, you'd get a couple of orders of magnitude increase in the largest possible bacteria, right? So these small contingencies would have huge effects on the possibilities for life on a different planet. Now, we can imagine a ribosome being much worse, and in fact, maybe this is the reason in lots of places that you don't encapsulate is because your macromolecules are too big or too inefficient. It's hard to pack them into a space. But there's also questions about how much better things could be. And here too, this sort of connection with physics gives us an answer. So we took the ribosome and we said, well, you can think about this as an abstracted computational process. And work that began in the 60s with Landauer pointed out that one can translate computation into thermodynamics and start to bound the energy requirements for any computational process that you might be interested in. And so what the ribosome is doing computationally is really just taking an unordered set of letters and writing those into one string. So that has a really simple entropy transformation. And we can bound then how much energy it would need per amino acid to do one of these transformations. And then we know how much energy the ribosome actually uses, and we can make that comparison to understand what the ribosome is doing. And here we find that the ribosome is 25 times less efficient than the ultimate physical bound. Now you might say 25 times off sounds pretty inefficient, but just for reference, if any of you who are taking notes, those notes are being stored to your computer right now at 100 million times worse than this bound. So even after almost 100 years of Moore's law, which is impressive, we're still very far from the sort of molecular efficiencies that life has achieved, which is interesting. But this tells us that it's unlikely we get a much better ribosome anywhere in the universe, right? We may have a ribosome that's about as close to the best that any string processing physical device can do, made of any material anywhere in the universe. And that's a really nice bound to know. It tells us that this sort of transition is unlikely, whereas this transition might be much more likely. And we have to ask questions about how hard it is to get a good enough ribosome that we can create encapsulated life. This sort of idea of asking what sorts of constraints are fundamental across the universe for living systems, we've reviewed in much more depth here. This is a sort of mega paper, a super group, to use a 90s term for music. This is sort of a super group of people thinking about these constraints, and through this, we go through basically all the ideas of information, energetics, so forth and so on, and just sort of detail what we might expect to be true for life anywhere in the universe. And I'll say that at a conceptual level, I think the sort of program that we're outlining here is to say that typically we think of biology as these very complicated branching evolutionary processes with lots of randomness and contingency, producing these diverse set of organisms. If we expand that to origins of life thinking or astrobiological thinking, one could even say those evolutionary trajectories might happen in fundamentally different materials. We might have material diversity across the universe composing life, and in each of those we have one of these complicated evolutionary trajectories. So now life is sort of not only unimaginably complex or diverse on our own planet, it might be unimaginably diverse across the entire universe if different materials are being employed. Some people would say there's more material constraints than we think. I know Nick would make strong arguments for certain sorts of chemistry being more likely than others and sort of restricting this. I think that's still a really interesting and active area of work, but for generality, let's say that materials can be quite diverse. Even if that's true, and even if in one material you have this vast diversity, we would argue that these physical constraints still compress that diversity into something that's understandable in this law-like way that I've shown, because those constraints are causing lots of different organisms and diversity to fall along single curves defined by a small set of physical constraints, dominant physical constraints. And so maybe this compresses life a bit more than this vast diversity that we've already thought of. And then the question is, across all those physical constraints, are there some sort of meta principles of optimization, of optimality, some sort of functional concept we haven't come up with yet that would compress all of these into an even more tightly bound space? So for example, very general theories of intelligence, and I'm using that in the Dennett way that would go all the way down to bacteria. Very general notions of intelligence might be something that's fundamental to life, and that would actually take lots of different types of physical constraints and compress those into one very small region of a certain sort of optimization principle that I think we're all still searching for. And maybe this is where truly universal life lives. Now I want to mention that the program I've sort of been laying out here is really sort of addressing different types of scientific cultures. So a few of us wrote this essay recently talking about in the biosciences, which could be extended to, in the biosphere sciences, which could be extended to just the biosciences. We really have different types of cultures operating with different goals and motivations. And what we really need to do is bring those cultures together. But it's also useful to recognize which cultures have been focused on and which haven't. So in this paper, we identify 3 cultures, what we call exactitude culture, variance culture, and coarse-grained culture. These top 2, variance and exactitude, are really the ones that have been focused on most intensely for most of the history of biology. To map that onto the physics language I was using a second ago, the language I've been using throughout this talk, variance culture would be this focus on lots of individual species. How many species can we document? Can we list all of the traits of all those various species? How much can we know about all of biology, right? Which has been part of the program of biology for quite a long time, and is important. We need that data, we need that knowledge to do lots of things. But it's focused on the variance, on the differences, and documenting that. Then you have this exactitude culture, which is the sort of physics I talked about with climate science or heliophysics, where you're really trying to take all the things that you know. In the physics case, that's all the fundamental physical laws that you know, and put those together into complicated models and simulations to predict things. What's interesting is that in a lot of biology, we've gone from variance culture to exactitude culture, right? Where we're trying to model the entire system as well as we can using all of the details we know about all of the species. And there are many biological models where people are really trying to put in thousands and thousands of species and model them in quite a lot of detail. So then the question is, what's this coarse-grained bit? And we would argue this is the piece that's been understudied in biology. In physics, it would be finding these fundamental laws, these coarse-grained representation, these sort of reductive, simple aspects, and then using those to build up to detailed models in order to explain lots of variance. In biology, I think what we're trying to do now is go the opposite direction and take everything we know about variance and find the systematic regularities that exist to get some coarse-grained laws and then re-expand to build better models and maybe to make other sorts of detailed predictions about living systems. So just to conclude, I think there are easy answers in biology. Some of those are even the answers we want. And what I mean by that is I'm not only focusing on saying, if we ask different questions about biology that have answers, but those questions and those answers aren't anything that any biologist would care about, then I've done something wrong. But what's interesting about the group of people working in this area is that the types of answers we're getting are doing things like systematizing diversity. They're addressing biological diversity. They're doing things like predicting the major transitions in the history of life, which is something we want to do. And they're helping us understand things about universal function, which makes it both more possible to understand the deep origin of life on our own planet and to search for life beyond planet Earth. And finally, I think if we do find some coarse-grained representation, some law-like features of biology, ultimately there may be questions in biology those don't directly answer, but they might be nice, interesting starting places to build up other theories that can do things like explain what's unique about humans, or why a certain species has a very particular toxin, or why the moth and orchid live the type of symbiotic lifestyle that they do. And so in many cases, these are just help us to create starting places that will make it easier to address the hard questions. And I'll leave it there, and thank you for your time.
Thank you very much to Sean and Jen for this invitation. And I agree with Chris's talk. I had a delightful lunch with him on Thursday, but I wanted to be a little bit of a devil's advocate that biology is, as he acknowledged, in many ways still it's harder, it's more complex than natural non-living systems. And I think one of the reasons that I see this is still, there's much harder questions to ask. Chris tended to ask questions about why the living systems are the way they are. And that, from my perspective in the biophysics department, the Biophysical Society, we tend to focus on how these systems function. And because of the combinatorial complexity within living systems, when you think about all the molecules they're made of, then that creates a vast search space to find solutions. And biology and physics are often taught in different ways. Physics is taught in terms of principles, biology in terms of special cases. And actually, I prefer the physics training, the principles. I think it's really important and we can find laws and universal trends in living systems in biophysics, but special cases are still also very worthy of our attention, right? So this is a cell, it turns into a multicellular human being. Why does this happen? I think Chris pointed out that at some point you can't exist as a small cell and there's natural selection, but how does this happen? And then further, this infant that is like a lump turns into a human that can play the piano and speak, and that this is absolutely fascinating and complex, and how this happens is really, we don't know. Although, especially for the first arrow, you can imagine, oh yeah, I can tell you that. So the cell divides. This is all encoded in its genome, and then it differentiates, and then you have different cell types. But in biophysics, we keep asking for more. And like I said, not so much like a small child who keeps saying, why does that happen? But for me, it's more like how does that happen? There's information that is encoded in the genome. It has to be duplicated, but there's physical processes. It has to, it's an actual physical set of molecules that have to divide and create two new copies. And of course, special cases matter also because, diseases are basically all special cases of these malfunctioning living systems. And I might like to understand, why we're here evolutionarily, but, I'm a very practical person and I am here now. So I care about how I can, cure my disease or more practically. And I think Chris does make this important point about questions can be easy or hard. I've posted this in my lab because I've become frustrated with I'm a scientist, my students, I feel like sometimes they don't know the scientific method. That my one undergrad said, we learned that in elementary school. I said, but you have to use it. And so what makes this hard? To me there's two parts that can become really hard. You have to formulate a hypothesis. and based on observations. And when I was writing a grant as a junior professor, this program officer said, what the biologists want, they want you to enumerate them all, every single hypothesis, so that you can test them all and explain the phenomenon. But so that's hard, right? That means that if you ask too hard of a question, you can't enumerate all the hypotheses and test them. So we keep having to ask narrower and narrower questions. That really tends to balkanize our disciplines into all these subfields. So, like, there's this whole subfield of molecular and structural biology. It's really critical, but they don't study living systems, right? This is the ribosome. So I use the ribosome because Chris included it. It's this multi-protein and RNA complex, and it's often described by the people who study it as beautiful. And I don't think it's beautiful. It's like it's a lumpy mass, right? It has like 70 or more different components, and it has no symmetry, which I'm actually going to return to later on. That this is part of what makes living systems so magical, is they function with this great asymmetry and complexity. Another way that things are hard is that you have to design an approach. So you might have a very clear hypothesis, but can you test it, right? Does the approach even exist? Is it theoretical or approximate what you're going to try to do with it? And this, but one thing that has changed is questions that used to be very hard are now easy, right? So back in the, I don't know, actually the 50s or 60s, we didn't know the structure of DNA or proteins because we didn't know how to solve something at the atomic scale. But now we do. This is a real structure from an experimental determination of a structure plus computation. So now we have even more revolutionary tools like cryo-electron microscopy. This is not a hard question really anymore. You give me a sequence, I can tell you. Also, we have AlphaFold. So we have neural networks that can predict three-dimensional structures. And but what I think is exciting about biophysics now is this allows us to return to harder questions about living systems, more about how you turn from a single cell, or how do you turn from something that's not living, like this ribosome, to something that's living like the cell. And so these are all the databases that we have now. We have all these macromolecular structures in this database. We have all these genomes in databases. We have machine learning approaches that allow us to predict structures. And we have all this information about the proteins and the RNA. There's all these molecules that are within our cells. There's literally not just millions of molecules in our cells, there are millions of types of molecules in our cells. And then what really gives rise to function is that they all interact with one another, and this can be determined through various approaches as well. So this combinatorial complexity is a hallmark of living systems that I think is really, it's distinct from natural non-living systems, like like a tornado or some other weather phenomenon. And so, and we know a lot of these building blocks. We know about the number of proteins, and these numbers may not seem so huge, but once you start putting them together, they can create these larger structures. They create this order, and that's where we get life. And one of the things I'm also really excited about now, that's also coming from the statistical physics community, instead of integrating out all of these details and trying to reduce to a small parameter space, which I actually think is a very powerful approach, and we do that as well. Can we also embrace this huge parameter space that these living systems exploit? And more now, people are taking advantage of the analogy to neural networks. So the deeper your neural network is, the more it can do, the more expressive it is. Right? These are the foundation models are like ChatGPT. What they can do is so much more vast than a small neural network. And a living system is just like that. I mean, in neural networks, every weight, every edge can have a different parameter. A living system, every interaction can have a different weight, a different edge. And every molecule, it's not just millions of water molecules, right? So it's not just condensed matter. It's hundreds, thousands of different types of macromolecules. There are all these different polymers that get put together. And so Andrea Liu gave a talk at the APS meeting, American Physical Society meeting, that was based on this idea of tunable matter, which can benefit from this analogy to neural networks. And one concrete example of something that we care about is macromolecular complexes. So I'm showing the ribosome here again. They are comprised. This ribosome has over 70 different subunits that come together. And then there's all these dynamic structures that form in cells, and they also are comprised of many molecules and many types of molecules. So living systems seem to be, taking advantage of this complexity of types. You can get from they have to start off from one molecule at a time and somehow get to their success. And they have to be designed, of course, not just to do the correct thing, but to not do the wrong thing, right? So you don't want it to form a partial ribosome. It has to get all 80 subunits together. I keep picking different numbers. And it needs to assemble this protein cage, not in the cytoplasm, but only on the membrane around the receptors. And they do this by taking advantage of this huge parameter space. And so to sort of complete this example, right, of a ribosome, actually, it has to be optimized for many things simultaneously. And this is, in some ways, this is what foundation models also have to be done. You can't, it doesn't just have to function, it has to get to a giant complex from all of these different subunits. It has to get from the nucleus where it's put together to the cytoplasm where it performs its function. So there's all of these different selection pressures on it, and having many distinct subunits most likely provides it an advantage for achieving all of these things. And so, in my lab, we asked one question: how does the assembly process, getting from a bunch of single things into this one macromolecular complex, place physical constraints on these macromolecular assemblies? And there's different challenges you can imagine, but one of them is if you have to get to this guy here in the bottom and you start off from all these singleton guys here, there's all these different paths you could take and that you have to take, right? These things don't instantaneously coalesce. They have to bind to each other. It's a physical process, local interactions. They have to meet and bind. And if they form all of the possible combinatorial intermediates, they'll never get to the ribosome. And so there's a lot of selection pressure, and we, show that there's constraints and that you can benefit from diversity in subunits just because it expands your parameter space. And so let me skip the Clathrin example. And so, are the questions in biology and biophysics getting easier? Certainly, our tools are getting better, and our data sets. And so, I'm excited that we can answer the questions that previously seemed impossible because you could ask them, but you could never enumerate all the hypotheses. So you kept having to narrow down. And so now we can go from studying these subcellular processes that are not alive to actually the predictive virtual cell. And that's it.